All righty. Welcome, everyone. Hi, welcome. We're going to slowly get started here. The first thing I wanted to do is go ahead and introduce our speaker. As people are kind of filing in to the webinar, I understand that we have about 5 people so far. We're expecting a few more at least, but we are going to go ahead and just get started with some of the pleasantries. So the first set of pleasantries are I will ask that people type in their questions to the chat window if that is okay. Because typically we will have a lot of feedback and other problems during the presentation if I allow participants to have microphone rights. So I will disallow microphone rights during the presentation, and then as soon as the presentation is over, I will go ahead and allow all of the participants to have microphone rights. However, if you have questions, I encourage you to write them into the chat window, and then we can ask the speaker and make sure that Dr. Hongwei gets through all of the questions as they come through. So he has also asked to keep the questions at the end. So that's it for the little background in terms of how the webinar will run. So thank you for attending. And I'd like to thank Dr. Hongwei Dong, who is an assistant professor at UCLA currently in the Laboratory of Neuroimaging. He started out in China in the 4th Medical University of, I believe it's Cheyenne, China. I could be wrong. I'm sorry with the pronunciation. Chiang. Okay, perfect. Thank you. And did a postdoc at USC, working with Dr. Swanson. So this is a well-known anatomy laboratory. Then moved up to a research assistant professorship there, and then moved up to Seattle to work with the Allen Institute for Brain Science. And this is possibly where much of the connectomic and high throughput science that Dr. Hongwei has since been working on. And then what Dr. Dong had gone back to UCLA across town from USC. I'm sure that there's some rivalry going on there. But in any case, Dr. Hongwei Dong will discuss a couple of things with us today, primarily centering on his work in connectomics, as this is a very big area right now, a very big area of study. Also, a lot of people don't really have a good grasp yet on what this kind of data can really tell us and show us. We've had some wonderful work coming out of this, including some exciting images from DTI and other things. But the question always with a new set of techniques and a new set of analyses is, can we interpret this data properly? And what are the sorts of things that it can really tell us? So without further ado, I will go ahead and mute my microphone and hand this over to Dr. Dong.
Hello, everyone. Good morning. All right, so I'm going to talk about this connectome project that is happened at UCLA, and many of you know that currently we have several different kind of connectome at different scales, human connectome launched recently by NIH. That's a $40 million project that mainly use in vivo technologies like functional MRI, DTI to look at neural networks in human brains. And on the other end, that is called macro level connectome, that is brain bone mice or array tomography or high throughput EM studies. And that kind of really advanced technology can allow us to look at connectivity at a synaptic level or a single neuron level. So our project here is kind of like in the middle. Is just in terms of their scale and also resolution is between this in vivo imaging, human imaging, and this macro scale level in the middle. And sort of like this project is kind of like continuous of um, one brain mapping project that has been started like almost like more than 2 decades. So I show these slides here. This book is mapping the brain and its function. Actually, it's kind of like a summarize for what happened in like 1991. They have one committee called National Neurocircuitry Database Committee that is organized by NIH and the NIMH, NIDA, and several agents, and include also NSF. They organized a number of really well-known great scientists together and talk about uh, that time for technologies in the neuroscience and also computer science put together, try to do this kind of large scale brain mapping. Actually, that was the time NIH already started try to think about um, this big project. So even for today, some of these committee members like Dr. Larry Swanson, David Van Essen, and Golden Shepherd, they are still around, very active. And we know that Dr. David Van Essen is kind of like he is major players in the Human Connectome project. And Dr. Larry Swanson, currently he is president for Society for Neuroscience. I think he is going to also put lots of effort to try to emphasize this Connectome project for understanding the brain circuitries. So I really like these words, and that's summarized in this book. So how we see really affects how we think. So that is, I think that's kind of like a principle for why do we need to do this kind of large scale brain mapping project to present this connectivity data in different formats. So and so I put these two sentences really is a pickup from the book. They put interpretation, complex perception, and even the appreciation of the beauty often begins with visual information. That's really it's like for we start to understand our neurocircuitry data and understand the behavior. And to understand it, we need more experimental data and new ways of assembling the diverse kinds of related data into an integrated whole. So that's pretty much like this kind of effort already for uh, 3 decades. And that was recommendation by that committee by that's almost 3 decades ago. And they recommend to construct this kind of a three-dimensional computer map and also this kind of models. And also that time they also already recognize we need to do this kind of a map in both normal and also different disease states. So and so we talk, this connectome term really is come from only a few years. That's Oliver Spahn to coin this term. And what does that mean? People have different opinions and different thinking. What does the exact means for connectome? I just pick up this sentence from a wiki page. That is, connectome really is a comprehensive map. I emphasize map here. And so basically, the maps connectivities and also show the interactions. And that is like an interaction, really, that is um, what I emphasize today in my talk. We're not only just look at the neuronal inputs or outputs, we also look at their interactions between these different structures and also between these individual neurocircuitries. In other words, we are trying to start to look at this neuronal connectivity at network level. And I'm going to explain in my talk what does that means for network, because like currently in especially in human studies, people have studied and uses really fantastic MRI, DTI technologies, and uses graphic theory to look at how different brain structures they correlate with each other and how that one relate to different functions and also in different disease state. I think this is really fantastic. And however, the limitations for this in vivo imaging and in human study are mainly two things. One is resolution. So we know that DTI, MRI, their resolution is not, you cannot, it's not single at the axon or neuron level. And also even for DTI, they measure this connectivity. Actually, it's mainly measured kind of like axonal bundles or pathways. However, the how this axonal, how this connectivity started from the neuron or terminate on their target, that all happen in gray matters. And in whatever happened in gray matter, so this synaptic connectivity, we cannot say it uses DTI or functional MRI technology. That's one thing. That's major limitation. And the second limitation, I think, is for directionalities. So when we look at human studies, these networks, and you see like a structure A connect to structure B connect to C, but that is based on the statistic um, calculation. It's not like a real hard wiring. You know for sure structure A project to structure B or structure B or project C. So in other words, whatever the networks we know in human studies, it's really important for us to validate in human and in animal models, use these tract tracing technologies. That's what we are going to talk today. And so in a, and also this try to map these neural networks is has been around for recent 5 to 10 years, and some large groups like Allen Institute for Brain Science and Cold Spring Harbor laboratories and several different groups, and they're just use different technologies to do it. And for us, we're just like one small regular academic lab, and we're just trying to use our strengths and try to do whatever we can. Um, I'm going to illustrate what's the specificities for our technologies. So the general strategy we use here is we try to systematically map neuroconnectivity of this mouse brain. This is a C57-6 black. Um, so the first question really is how can you, in a limited time and with a limited people, to map the connectivity of the entire brain and produce some kind of useful data for people. So at the beginning stage, we designed this strategy called grid-based injection strategy. So in that case, we divided half of the mouse brain into these 1 millimeter cubics. And our thinking is we can systematically make injections into all these little grids and either aim the center or corners. And totally for the half mice brains, about 200 these kind of cubes. And so for in that case, we can quickly to do a quick survey for the connections for the entire mouse brain. So that is one general strategy. And later on, we realized some flaws for this kind of a strategy because for neuroconnectivity, in order to get something really useful data, you have to be really careful to compare your injection site with around other structures. So in that case, you can make sure that your connectivity really is associated with one specific nucleus, or they come from the contamination around other structures. So we optimize our strategy. Really is we call it kind of like combinations of anatomical based and grid based injection approach. In that case, we focus on some structures and we do systematic, very detailed injections. And so in that case, we can really make comprehensive data for one single structure, and then we move along from structure by structure. For example, we start from olfactory bulb, and then we can look at the cortex, amygdala, hippocampus, thalamus, all the way to brainstem. So that's the general strategy. And so this picture just show you for like a kind of like a three-dimensional view for the mouse brain, and construct based on the atlas I did for Anna Institute. So you can, from here, you can see the kind of like a 1 millimeter cubes for the brain structures. And so what we do is for one mouse brain, we make two injections. This technology really is optimized neural tract tracing technology that has been used by for the last 3 decades. That's kind of a classic neural tract tracing method. However, we optimize them. And so basically one brain, we make two injections as we show here, one pink, one yellowish. And for one injection, we have mixed tracers, one anterograde and one retrograde. As we show here, these slides here, the first injection here, that is mixed BDA and fluorogold. So BDA is going to pick up by neurons and then send to their axon and label their axon terminals. And a fluorogold is going to pick up by axonal terminals and then retrogradely back label their upstream neurons. So the same thing also is for PHL and CTB. And PHL is anterograde tracer pick up by neurons and label their axonal pathways and also their terminal butons. And color toxin or CTB here, that's a back label their upstream neurons. So the really advantage to have these two injection and 4 different tracers are, we can arrange, if we arrange well for their injection site, we can look at their parallel circuitries, and also we can look at their interactions. So in my later talk, I have very good examples to show the power of this technology. All right, so here I show the one examples of our injection site. As if you can see on this picture on the right side, that is a really bright spot. That is the injection site in anterior insular cortical area. And then you can see this green axons from injection site cross corpus callosum, go to the contralateral site, and that in that red box, you will see that that's really dense labeled axon terminals labeled by PHL. But at the same time, remember, like our one injection site is mixed tracers of the PHL and color toxin. So color toxins label their upstream neurons. As we show this on the bottom left side, and these pink neurons, that's the color toxin labeled neurons. In the same region, intermixed with the green axons. And these pink neurons, they project back to the injection site. So in that case, we can label both their outputs and also their inputs associated with one single injection site. And as we can show here, if the retrograde labeled neurons and the anterogradely labeled axons, if they are intermixed, which means this spot here, they have reciprocal connectivity with the injection site. And here, this picture just show you another examples with 2 parallel injection sites, and a greenish with pink in the middle, that is the PHL color toxin in secondary motor cortex, and the BDA and fluorogold, that is in the ventral anterior cingulate area. And you see these 2 injection sites, small injection site parallel. And on the right side, this picture is just one example sections through the thalamus level. And as we can see here in the thalamus, there's one kind of like a busy area. And if you look at the bottom pictures, that actually that area, you got like the first thing you will see the red color and green color, 2 parallel axonal terminals. Green axons come from secondary motor cortex, red color axon terminals that is come from ventral anterior cingulate cortical areas. They next to each other. You see they are parallel, but they are not intermixed. And on the another picture, you'll see that green axon intermixed with pink neurons. That basically these pink neurons project back to the secondary motor cortex. They associated with the same injection site, which means these thalamic nucleus, here is the posterior thalamic nucleus, they have reciprocal connections. But at the same time, I want you guys to see that these green axons actually intermixed with these fluorogold neurons. It's on the bottom right picture. The green axons intermix with these yellow cells. These yellow neurons, they project to the secondary, project back to the ventral anterior cingulate cortex. So from this sense, the conclusion, the message here we got is, so secondary motor cortex, MOS, project to this thalamic nucleus. Here's central laminum CL labeled as here, and also lateral medial dorsal thalamic nucleus. And then this nucleus project to the anterior cingulate cortex. So this work kind of like a two synaptic circuitries. And on your right side has, on the left side has two other pictures labeling. You can see the multiple labeling in the cortical areas. And here's more enlarged pictures. The first one, A, that is the PHL intermixed with the color toxin neurons, which means this thalamic nucleus has bidirectional connections with their injection site. And the second one, B, shows BDA labeled axons. And C and D just shows a PHL, CTP labeled neurons and fluorogold labeled neurons. And as you can see, the fluorogold is clearly in different layers of cortex. But at the same time, I want you guys to pay attention on C, that's a pink neuron on the background of their nasal staining. And this nasal for all the neuroanatomists is very important because the nasal can give you actual background, actual cytoarchitecture. So it will tell you exactly your labeling in which structures. And on your right side, labeled as E1 and E2, that is really high magnified pictures to show. Like once we see this kind of labeling, and we can always do the confocal and to look at high resolution. If we can see this axon terminal buttons in E2, form this kind of a close opposition on the single neurons, which means this area potentially has synaptic connectivity. All right, let's move on. So this several other sample example pictures. Like here we can show two different population neurons labeled by different tracers in thalamus associated with two different cortical areas. And here that is two injection site in two different parts of a somatosensory cortex. You see the green axon terminals and also red axon terminals associated two different injection sites. And here basically is another pictures in thalamus to show the reciprocal connectivities. And these two clusters, several clusters, intermixed axon terminals and neurons, which means these several thalamic nucleus, they have this reciprocal connectivity with their injection site in cortex. But at the same time, you see like on your left side, only this area only has PHL labeled axons, which means this thalamic nucleus only receive inputs from cortex, but it doesn't project back to the cortex. So this kind of information, unidirectional or bidirectional connection is kind of like important for we understand the function and also behavior. And here's another picture. So shows the intermixed axon and neurons in the cortex. So anyway, okay, and we can have at the same time show four different tracers. That is to show these pictures here: green axons, red color axons, and pink neurons. And actually, they also fluorogold labeled neurons is yellow, kind of like intermixed with red color axons in the middle of thalamus. Just because here you cannot see it, but if you turn off other layers, you will see that. And after we produce this data, we do informatics process, and our goal is basically present all our raw data online. So that will be as kind of open resource for general public and also neuroscience community. And here's like one screenshot of our website. And currently we have like more than 70 cases, but we have more cases we need to process. And it just this kind of a work actually is more labor intensive to process data put online than the producing the data. I don't need to go to detail why it's so labor and time consuming. So when you go to our website, one important information I think is for each case they have one little magnifying glass. So it shows the actual injection site because our actual injection site is small. And however, in order to show the axon terminals and the picture itself overexposure. So like as we can show here, injection site actually is quite limited. And on their background, that is nasal staining. So give you kind of accurate information. You know exactly where injection site are. And then here's another sample injection site. You see the axon cross corpus callosum go to contralateral side of anterior cingulate cortex. And actual injection site should be much smaller than this. And so here's I just show you like our iConnectome visualization tool. So when you pick up the case in our site and you will see the injection site, and then you will go through each different sections. On your right side, you see that little windows has little icons, and you can pick up the tracers. You can, for example, like the picture here shows the green channel. That's a PHL labeled axons. But at the same time, if you change the opacity and you click the little icon on the this little window, you can see the combinations of different tracers. For example, here, if you change the opacity of the green channels, you will see underneath of these pink labeled neurons. And so in that case, you can see their actual spatial correlation for these different tracers. And another important thing is underneath of all these four different tracers, we have their actual nasal staining and for each individual sections. So this nasal staining can show you exactly cortical layers and also different structures. And you can see here, when you change the opacity, you will see the neurons on their actual nasal staining on the top of their cell architecture. And so this at the same time, you turn on three tracers, you see their distribution patterns. Actually, you can see their single neurons in exactly which layers and corresponding to their nasal staining. And so this kind of information really is accurate to help analyzing the data. And on the other hand, basically on the windows, like all the our images register onto the Allen reference atlas. And this atlas just give you the actual anatomical reference to tell you your labeling or which structures these labeling are. And so you can show this one in parallel. And in another window, you can see, as what I said, you can change the opacities, see this labeling on the top of their set architecture. But at the same time, you can also see your axons on the top of the atlas. So when you look at these little windows, you see on the top that is called atlas, that is actually is on underneath of all the tracers and also that nasal staining. So when you change the opacity, and you will see your actual labeling on exactly the top of the atlas. So this kind of a principle really is just like kind of Google Earth map. So pretty much like just like a Google Earth map, you have this map, or the Google Earth, you have map on the top of satellite pictures. That's what we are trying to do it in the future. We hope our map can really provide kind of like this Google Earth like connectivity map and also this GPS roadmap to show people exactly give any um, two structures, how they link to each other directly and indirectly. give you kind of like a driving direction map. All right, so that is like a general strategy how we produce this data. So on the other hand, because we are kind of like our goal really is not only just map the inputs output of each individual structures and our strength really is try to help people provide kind of like a more useful information for people study the behavior, study physiology, study function, and also study the disease. How this neurocircuitry, neuro connectivity data to help people understand for their own research. So our strength really is like analyzing our data and then try to start to characterize neural networks and to help people understand so at networks levels how different brain structures interact with each other. The reason for that is, as we know that like for complex cognitive functions or behaviors really is involved multiple structures, multiple circuitries. And like as we show here, that is for zebra, they like you have when they eating kind of like an ingestive behavior. However, one line approach, they have to rapidly turn off their eating behavior and wrong way. So basically, it's a rapid switch off between different behaviors, and this absolutely is involved multiple brain structures and at least two independent neurocircuitries. And as we know that like complex behavior, when you dissect complex behavior and to the bottom really is motor neuron pools in spinal cord, and that is control the individual muscles for the muscle fibers. So basically for somatic motor neurons innervate different muscles. At the same time, autonomic individual ganglionic neurons that's also controls the sympathetic, parasympathetic activities. And the same principle also for neuroendocrine. So basically, for all these complex motivated behaviors, they have to be like integrate between these somatic, motor, autonomic, and neuroendocrine activities. So the question is how our brain organize this different motor system for different behaviors. For my point really is just for like the whole brain organization, really just like the piano. Like we know that the piano has only a certain number of the keys, like the numbers of the keys are fixed. However, the combination is different. So when you have different combinations, you can really put it together in to like almost like a numerous piece of different music. And that's basically the way of the brain organized. Like for our job, really is try to find out the organizational principles in which way, how different system, different brain structures, if how they organize differently for different brain function and behavior. And so in order to do that, like if we look at the bottom, basically two individual cell bodies, neurons, we're trying to look at how neuron A project to B and B and then send the axon terminals to its downstream other targets. And neuron A, they also receive inputs from its upstream other neurons. So basically, if we put together as a little network, this recently Larry Smith and Mihail put together this kind of this paper, kind of like a hypothesis model. Really, is if we try to look at the different individual brain structures or can be individual neurons. Each one of them has their own individual circuitries. However, how they interact with each other and does their circuitries parallel, segregated, or how they interact with each other. That is our job, our goal for our Connectome project. And so basically in the next, I give several examples how our work and construct different networks. And we look at topography, segregation and interactions between networks. So the first one is like we recently published a paper in the Frontiers in Neuroanatomy. For that one, just like as a kind of beginning stage, we try to look at the neural circuitries of olfactory bulb. Main olfactory bulb, people have studied that one for like almost like over 100 years. And we're not only just repeat to look at olfactory projection pathways. That one basically people has known that for a long time. Um, However, use our strategy, we make very small injection into this different part of olfactory bulb. As we show the picture here on the top um, medial side, or the bottom side, medial side, or the lateral side, and we look at exactly their axonal projections, and then we find out actually their axonal projections in the lateral olfactory tract. These axons come from different part of olfactory bulb. They are segregated, and they with clear topographic patterns. In another way, so on the top medial side main olfactory bulb, their axons in the lateral olfactory tract is pretty much more like a dorsal lateral side, but their bottom from the medial um, ventral parts, their axon, like pink, is more like a ventral medial side. So that is basically look at their axon topographic patterns. On the other hand, because we have these double co-injections, so we find out actually if our fluorogolden injection site only involved granule layers of olfactory bulb, we got labeling neurons is only in the posterior part of a piriform cortex. And however, so if the injection site involved more superficial uh, layers for the olfactory bulb, and we don't get the labeling in posterior part of piriform. And so in order to confirm that one, actually we made the PHL injections into the posterior piriform cortex. We find that their axon projections really is kind of in the deep layers of granular cells. As we show here the pictures on the second row, that's E. You see these green axons just right in the center of the olfactory bulb. And at the same time, if the PHL injections into more anterior part of piriform, you will see the axons actually is on the superficial granular layer. And at the same time, also they has a direct projections on the mitral cells, as the bottom picture we showed here. So, and especially we find here is like from piriform direct projection to the mitral cells. It turns out actually that is kind of like a novel thing. And based on that, we generate a kind of like a hypothesis models how the piriform connect to with the main olfactory bulb, because like currently seems like current dogma seems like is olfactory bulb project to piriform cortex, but piriform cortex project back only to granular cells. But what's the novel finding here is like first we find the topographic patterns like anterior piriform and posterior piriform project back to olfactory bulb, they are different. And the second thing that we find is the anterior piriform cortex not only project to granular cells, but also directly target on the mitral cells. So that is one study. And another study here is kind of like continues like the work. People always question about the dorsal and ventral hippocampus. Are they functionally distinct structures? And lots of behavior study has done that. And we think based on the their gene impression patterns, we find the dorsal hippocampus and ventral hippocampus, their gene impression patterns really is different, which means their functionally should be very distinct. That is supported by numerous functional networks. And so to confirm that, we made two injections at the same time in the same animal, dorsal hippocampus and ventral hippocampus. And we showed, here's one example pictures, and you at same pictures, you can see four different labeling. For example, here you see the green axons labeled in hippocampus. That is come from that dorsal speculum there. But at the same time, these pink neurons in the dentagers, they project to that injection site. And on the bottom side, you see on the right side, that's amygdala labeled red color axons that come from ventral speculum C1. And also you can see a few yellow neurons. That neurons project back to the hippocampus, ventral hippocampus. So if we show here in larger pictures, you will see that this 4 individual labeling, and which means 4 different pathways. They are completely segregated here. And so this is another pictures. And especially amazing is like you can see like the green axons and red color axons. One is come from dorsal speculum, one from ventral speculums. Like they are labeling in entire brain, almost like completely segregated. They have no overlap. So that's very impressive. All right, so so that is to look at parallel circuitries. And another really strength for our technologies, so we can really look at multisynaptic neural networks. Like for example, like what I showed here, like we have 2 injections also in the same animal. So one injection, the BDA and fluoro gold, that is in the dorsal striatum called a putamen, and another injection is in the cerebellar nuclear. Here, as we show here, is a green color, that's PHL injection. But remember, like our double co-injections, we also have PHL, so we can label upstream Purkinje cell neurons. They project to the injection site. And secondly, we look at in the thalamus, we saw the green axons come from cerebellar, this deep cerebellar nuclear, and same time this green axon target on fluoro gold neurons. The fluoro gold neurons actually is project to the dorsal striatum, and then at the same time we can see the axons come from the striatum in the substantia nigra. So from this case, we can really look at this the kind of like cross 4 synapse, 5 stations, this network. So if you can imagine, you can do this kind of a different combination, put the injection site in different structures. You will see this kind of many numerous these multisynaptic circuitries. So finally, I want to just do the last one, the little complex. We want to address some more complex networks. So people have studied these interactions between the medial prefrontal cortex, amygdala, and hippocampus, because the interactions between these 3 structures are really important for this all emotional and cognitive related behaviors like fear, especially like people have studied fear circuitries. So we're trying to understand at next level, high resolution detail level, how they interact with each other. And so the first ones, we put the 2 injections into the paralimbic and infralimbic cortical areas. And these 2 cortical areas next to each other, their connectivity in crossing entire brain. As we show this diagram, it's really complex. They are very distinct networks. They are not the same. But we focus on how they connect to this called basolateral amygdala anterior parts. And as we show here, this BDA axons that's come from injection site in the paralimbic cortical area. And these axons you see on your left side on the top, and this BDA labeled axons. The same area also has fluoro gold labeled neurons. So which means this part of the basolateral amygdala, they have this bidirectional connections with the paralimbic cortical area, reciprocal connections. But at the same time, we saw these green axons come from infralimbic cortical area. However, interestingly, these pink neurons, they project back to the infralimbic cortical area. They are not co-localized. They are not intermixed. Instead, these neurons, they are outside. So in that case, infralimbic cortical areas and this medial prefrontal and medial and this basolateral amygdala, they're also bidirectional connection. However, it's not like a PL and the basolateral amygdala. They're direct reciprocal. Here's bidirectional. So basically, lateral side of the BLA project back to the ILA, but at the same time, ILA acts on terminals, more terminal on the medial side of the basolateral BLA. So if we look at more higher resolution things more interesting is because in the injections are two injections in the same brain, we can see the axons come from inferior limbic cortical area, actually intermixed with the floral neurons. It projects back to prelimbic cortical areas. So which means actually the circuitries ILA go to this part of basolateral amygdala, and then this part of basolateral amygdala project back to the PL. However, the neurons project back to ILA is outside of these terminal fields. So it's kind of a complex network. So to confirm that findings, so we made the injection into this basolateral amygdala. So after we make injection here, and then we will see the back labeled neurons in the medial prefrontal cortex. If we look at more high resolution images, what we can see here is like a green axon and pink neurons, they intermixed. That is here is the prelimbic cortical area. At the same time, the neurons, pink labeled neurons, that is project to the basolateral amygdala. However, here you don't see much axons. That is confirmed our results in last case, and then we can construct these complex networks. All right. So yes.
So I'm gonna kind of ask you to either start to wrap it up because we have a question from Gully, and your time is kind of coming to an end here, and I'd like to get some questions because I think we're gonna have several others. So
okay. Let me just wrap. Yeah, in
perfect.
2 minutes,
Thanks.
just 2 minutes. Yeah. So anyway, so the rest of pictures just show this kind of networks can be really complex, and it's a multisynaptic networks, and that really help us for understand these complex brain functions. And at the same time, like the picture I showed here really is we can come to the single neuron level because two different injection sites we can see intermixed neurons in the same area of the basolateral amygdala. However, this we can see here is two neuron populations. They are intermixed, but they are not colocalized. All right, so this is the same thing. So in that case, we can really construct these complex neural networks, and this hypothesis model can be tested by other people using other technologies for their behavior, physiology, and functional studies. And in the end, I just want to thank all people has done the work, and especially here like Brian, Hody, and they did a huge amount of work here. And also we have our neuroinformatics team, and really several senior scientists, Ars Toga and Larry Swanson and Harvey, give us really great suggestions, advice. And thanks everybody for your attention. Okay, I can stop here and take some questions.
So I think while I'm adding microphone rights back to the folks, back to the participants, Gully had one question. Gully, you should be able to turn on your microphone if you would like to ask it specifically. But the oh, you can see the question there.
Yeah, I can see the question. Yeah.
About the olfactory bulb.
Uh-huh.
And I have a couple of questions also. So, but we should probably address the questions from the audience first.
Okay. So Gali, should I just go ahead to answer a question? Okay. Hey Gali, how are you, man? Okay, so to answer your question, actually, that's exactly what we want to do. So based on our neural circuitry, that hypothesis model, so we predict, so from posterior part of piriform cortex, they project back to the main olfactory bulb, target on only these deep granule layers. So in that case, if you stimulate posterior piriform cortex, you should be able to only inhibit the olfactory bulb, the mitral cells, because that granular cells are GABAergic neurons. So if you excite the posterior piriform cortex, you should be inhibit the mitral cells in the main olfactory bulb. But anterior piriform cortex, because they have direct projection onto the mitral cells. So in that case, if you stimulate anterior piriform cortex, you should see the excitatory response for the mitral cells in olfactory bulb. That is one thing I think more directly to look at the predictions for their physiology. But like people, because I'm not in the field for the olfactory, and so I hope in the field other people work on the olfactory, they can test these models. They can do their own for their own behavior studies. Yeah.
Thank you.
All right. So any more questions?
So Gully's typing, but I can. Oh, Gully, would you mind muting yourself when this is the feedback that I was talking about in the beginning. If there are multiple microphones on, this just gets kind of crazy. But so I have two questions. First of all, thank you so very much. This is absolutely beautiful work. Thanks, Gully. So you know, obviously this data, and it's very valuable. It's wonderful that you are sharing this data and making it available for the community to take a look at, kind of pre-processing, pre-publishing. Really, it's a way of doing science that you know, I think is pioneered largely by larger projects, including, of course, the Allen Brain Atlas, or the Allen Brain Institute, rather, which is, you know, in my little opinion, absolutely fantastic. But it also brings up some very interesting questions. And so I wonder if you could speak to, while we're getting other people to put their questions in, the availability of the data, if you can also to the interoperability of the data with other projects. So obviously there are large projects, the Connectome projects and what have you. And so those are the kinds of two things that I really have been thinking as you were talking, not just that the data are beautiful, in fact, that they are extremely beautiful, but the question really is, how can one lab create an entire connectome? I think the answer is it cannot. It must be that you, even, you know, even with the wonderful work that you're doing, I don't know that you will come up with everything that there is to know about this. So at some point, we're going to have to put this data together with the other data. So can you talk a little bit to letting us know, basically, you know, what your ideas about interoperation with other projects, with other labs, look like, about how you think that this data will be interpreted by others, and
Yeah. Okay. So actually, that's exactly really great point. And from beginning stage, I thought about, I've been thinking about these questions. Like, you know, also like yesterday, I read the book. I have one book that edited by Georgia. That's 2005, and that is computational neuroanatomy book, and talk about its database, all kinds of stuff. That's really and also another reason I want to do this talk really is I want to start to build up more communications with like NIF and INCF and other groups, because as what I showed here, actually for us, it's easier to producing this data. We collecting the data, producing this data, and we can present this data online. However, the next really big step really is the first is annotation. Annotation is a huge amount of work, and I don't expect like my lab can do all of this annotation. It just takes too much time, and we cannot do it. It's just not feasible just by the one single lab. Second thing is to understand this data, and it really require we build up this database. That's need to be need a community effort, need into collaboration between multiple groups, and so we can put it together into like in the future really build up a kind of like a sophisticated database. So in that case, we really need to collaborate with many different groups. And another thing is like how to coordinate with other project like an institute, their connectivity data, and also Cold Spring Harbor Parts and Mitchell's connectivity data. One good thing is because all of these three projects, we all use the same standard brain atlas. That's Allen reference atlas. That's what I constructed for Allen gene impression project. So we all mapped on the same atlas. So in the future, people can to correlate this data for example, like I have injection site in the somatosensory cortex, Allen also has, or Parsa also has their injection site. So we can look at they can validate each other. That's one thing. Second thing is like each project has their own strengths. Like my project has strengths, has both anterograde and also retrograde labeling. So you can look at upstream and downstream. Allen Institute has a great three-dimensional things. The way of their collecting data. So this just give people really all these open resource. I think give people multiple choice. But however, I don't know, like need some like a more senior scientists to organize kind of group effort to annotate this data. That is like really in the future, if we want to make all this data useful, we have to put effort together to build up the database and to facilitate people to use it. Yeah.
Yep. Thank you. I think that Giorgio had an additional question. Giorgio, you seem okay.
Can you hear me?
Yes.
Excellent. I really like Hongwei your discrimination across scales. So you mentioned, synaptic connectomics, and then brain voxel connectomics, and you rightly pointed out this is something in between. And I think that this is key. And one challenge ahead is to, in fact, relate these various scales to each other, which kind of must be done at a certain point. So I guess the first question is whether you are building in some aspects as to how this data eventually relates down to the synaptic connectomic level and to the brain region level. The other, I guess, question, it's more like a possibility than a question, is to find a way to extract statistical patterns so that the connectome would not necessarily have to be described at the level of individual pieces of data, but rather at the level of microcircuit or microcircuitry that could be, perhaps patterned throughout the cortex at least, and perhaps other areas as well. That's not, that's not really a question, but you probably have had some thoughts about how to distill the data that you're putting together.
We need more
framework.
powerful informatics tools, basically, in order to quickly analyzing this data. So in addition to manual work, so if we can have more